Hello everyone. In this tutorial, I'm going to go over our end-to-end cell analysis pipeline and how you can take advantage of this to simplify your cell analysis, especially yeast analysis tasks. So the first step is to put your files, your TIFF files, or the movies that you record from the microscope into the correct folder structure so we can analyze them. So you want to first, as part of your NAS folder. In this case, I'm in Sam's folder, but it can be in any one of yours. You can go ahead and create a folder and label it with the name of your project. Then inside of that folder, have another folder called data. Make sure this is spelled just like this, data. And then inside of that, we also need you to create another folder called IMStack. You can ignore, you don't have to worry about the other three here, but we just need this I capitalized, lowercase m, and then stack, and S is capitalized. Make sure it's spelled this way, and then inside of that folder, then you can put all of your TIFF files. Okay, and in this case, you can see we have two movies, and for each of them we have three channels: C1, C2, and C3. Then once you have finished this step, you need to go up here and you can open in terminal, and then right here you can actually see the exact location. So all you have to do is just highlight this area just like this and right click and copy. Okay, and so now we can then take this and put it into our software as well. Before we do so, however, we also want to have an output folder for all the analysis. So in order to create that, we're going to go back. In this case, I'm going to show you the folder I've created for myself. So Sina, here we did, I think I did final test. And so that's all you need, right? You just make a folder that includes all of your tests, and now all of the data is going to be analyzed inside of those. So again, you want to go back. So final test, right? You want to click, in your directory, create a folder for the output, go inside of it, and then go up here, open terminal. And now again, you can see the directory. I mean, of course, you can just type this out yourself, but this is easier. So you just copy it. And so now you want to have both of those, the input directory and the output directory. Next up, we're going to go to the software, and we're going to see how that's going to all work out. Welcome back. So now we can use the software to start analyzing. The first place you want to go is segmentation. So now, what you want to try and do is go on, this is our menu, so I'll walk you through it each step of the way. In this part, all we care about is this option, Upload Movies, which you can press, and it will open this panel for us. Now, there's an easier way to do it. You can just press D on your keyboard, and it automatically opens it up. Now here you can see that we can put our raw movie directory as well as processed movie directory. So for this one, we're going to take a look at the one that I showed you earlier. And then for the processed movie, again, same one that I'm showing you earlier. Make sure to also include this final backslash as well. It's optional, but I recommend it just in case. So you can just copy paste those directories that we got from the terminal right into here. Then go ahead and create your new project. So in this case, I'm going to say tutorial project, and you can put a description in it. So the files for the tutorial. And then you can go ahead and press submit. So once you press submit, you're going to receive an email notification letting you know that your movies have begun processing. Let's take a look at that next. Once you click the submit button and begin processing the movies through the server, you will receive an email with your name letting you know that we've begun processing your movies in your folder. And it also will tell you if there are other people that are working on other tasks using the server, so you kind of have an expectation of when we will get to yours. Once the movies have been processed, we will be able to send you another email letting you know that they finished and how long they took. So if you then also go to the server, you will be able to see that we are processing, right? You can see it's happening live. You can see it's already started doing a few different ones. And this is how it begins, right? So initializing Python, we are finding your fluorescent channels. We are finding a number of movies, and we begin aligning them. So now you can see we're at the generate cell mask stage. So once all of that happens, then you will receive another email letting you know that your movies have been processed. So here we can see we have 12 movies, which is basically 2 different movies and 6 traps inside of them. So 12 traps in total. We do 1 trap at a time, so we consider 1 trap to be its own movie. And then it also tells you how many minutes it's taken for this to be processed. Once all of this happens, you can begin tracking right here. And when you click it, you will be able to go to the website and start tracking it, which I'll show you next. Now that the movies have been processed, we can begin analyzing them. In this case, we're starting with segmentation. Although this is optional, I'm still going to go through it to show you guys the capabilities. For the segmentation page, the first thing we want to be able to do is to load our movie. You can see here we have instruction, no movie selected, press A to view all movies. We can also navigate to all movies with this button right here. Of course, we can also press A to open and close. In order to see your movies, you need to first select the user. In this case, I'm Sino2. And select the project that you had created. You can see the tutorial project here, but those movies have not been fully processed, so we're going to use a different project. And now we can see them all loaded up. We can see here the name of the movie, who uploaded them, and when. And if we open the trap list, we can also see a list of traps. In order for us to do segmentation, we need to select one of these traps at a time. So let's go ahead and look at this one. Movie has been loaded. We can scroll to get started. So we can scroll and scroll and scroll, and we can see the movie play out. It's a very intuitive interface. Of course, we also have forward with our keyboard, which is right arrow, and backward with left arrow. So we can also play out the movie by just holding the right arrow key or the left arrow key. Up here, we can see the movie name, the channel. We can also see the trap number, and we can see the time point that we're currently looking at. So let's get started with segmentation. Let's go ahead back to the first time point, which you can see right here. And what we want to do is to generate a segmentation mask for this image. In order to do so, we can just click on segment. Draw over the cell. And now we have both a boundary and a filled mask. If you want to also do this for daughter cells, we can. All we have to do is just create daughter, segment it, and this will be in blue. And now we can look at either the boundary or the filled mask. We can also look at the mother cell. And so with this approach, you're able to quickly generate these masks and save them into your NAS folder for future reference or if we need to train better machine learning models. You can also do these with different frames of the movie. So now we've moved on to the second frame and we can just segment again. Draw over it. And we now have our filled mask one more time. So this is segmentation. And next we'll take a look at tracking. Okay, so now we can move on to the next step. So the best way to navigate the software is go to howlab.webflow.io. For segmentation, that's where we were last time. So we clicked on segmentation and we were taken there. Now let's do prediction, where we do the tracking of the cells over time. So again, it's the same interface, very similar. So we press A, and we now select our user. We select the project, and you can see that we have our trap list here. And these are the different capabilities that each trap has. We can look at the different channels and see how they look. We can see the status, so whether it's in tracking, whether it's processed, and then whether we want to delete this trap, maybe the data is not correct, we can just delete it. And we can, of course, also extract out the CSV files. Once the data has been completed, we can also see how many frames does the mother cell live and how many children it produces. So we can take a look at that once we do the tracking. So in this case, let's select this one. And now that the movie has been loaded, we scroll to get started. So here you can see that it says null, mother, no centroid is found. Click on image to update the centroid. So you can see the mother cell, right? And if you want to track the mother cell, which in this case is this cell right here, all we have to do is just click, and now it's labeled. And we just have to click on track. And so now it's going to start tracking. And once the tracking is completed, it tells you the centroids have been updated. So now we just scroll and look at that. It's tracked it all beautifully with no problem whatsoever. And it usually will work this well. Sometimes you might need to update it. So if there are certain positions where you feel like it didn't do a good job, let's say in this position, it's not done a good job. You can see down here it says predicted, right? So that means this is a prediction, not an actual label. So all you have to do is just click it again, and now it's labeled. Once it's labeled, you can press track. And of course, it's going to track it one more time. There we go. There are more functionality that are embedded in this part of the product, and so we'll go through some of the other ones. The next step would be to track the daughter cells. So in this case, the best way to navigate is using the arrow keys. So let's keep going until we find a good daughter. So we can see one daughter being formed right here. All we have to do is just select the state, which is budding, and click. So now we have our daughter. You want to hover over the button. So once you hover over there, you can see it's labeled, and you can see it's the first daughter, daughter 1. In this case, right, you can see we have the daughter, and all we have to do is just click on this button, and we will be able to also track it. Now, the daughter tracking is not as accurate as the mother tracking, and so let's see how well it does here. You can see in this case it didn't do a good job. it was able to only predict it for 2 frames, and both of those are incorrect. And then after that, it's null, as you can see here. So the best approach is to just go one by one and correct it. So now we can kind of see it's here, and now we have another daughter that's being formed. So we can now go back and see how its position is moving. So we'll track it up until here. Now, if you want to select, if you want to add another daughter, all we have to do is, again, hover over our mother cell, select it, and let it know that it's budding. And so now we can just click anywhere. Before it was blocking our view. So all we have to do is just select our new daughter and click anywhere, and then this dialog follows it. So now we can come down here and put it just at the right spot. And we'll do it again for this frame. And you can see it right there. So let's keep going. We now have again this one, and this one, and this one. So now let's try to see if it's able to track. Again, if not, we can just do it manually. Look at that, it's able to predict it. So you can see this was labeled, and now these are predicted. So it's doing a decent job with this one. And now we have even more that are getting budded. So let's say budding, and we have now this item right here. And so you can see if we go hover over this, it follows what you're selecting. So if you're hovering over the purple daughter, the third daughter, then it will select the third daughter. If you're hovering over second daughter, it will select. Or if you're hovering over the mother cell, it will also do so, and it will let you know if it's predicted, null, or labeled. In this case, it's labeled. And it's actually, so you can see right here, it gets washed out, right? So right here, it gets washed out. And you can also label that. So you can just say washed. And then now, after that, it becomes null because it no longer exists. And you can see it's become active, and we have this kind of washed icon for it. But if you go back, right, it existed for just a few frames, right? So let's do this and this. So we can see it existed for just a few frames, and once it gets washed out, we also were able to detect it. Now this other daughter is still going strong, and now you can see there's a mismatch. So we can select it again by hovering over it, and then keep selecting it. You can see it's making a mistake. So this one was labeled, let's fix this one, and I think the rest are going to be good. So this is predicted, and now after this, it was not able to track it. So again, we can just label it ourselves until it gets washed out. So you can see we can keep going, right? And once we're done, so let's keep going here. So at some point, the mother cell will stop reproducing. So you can see it's starting to get old, and it stops reproducing. So once we get to that stage, all we have to do is just say that it stopped reproducing, which is, in this case, either it gets washed out of view or it dies. In this case, it died. So we'll say it died. It will ask us whether we're sure, because if we're sure, that means that this movie is now completed, and we're going to be moving on now to the next movie. And in this case, the next movie is XY2. Before was XY1, and the first drop of it. So it's able to understand, it goes from one movie to the next and has that capability built in. Let's take a look at this movie now. So this movie, let's try and track it. So here we can see it's tracked it very well. Now I'd like to show you a few more functionalities regarding daughter cells. So here we can see we have a new daughter that's being born, right there. So here it's budding. We're able to select it again, select it again, select it again. So now let's try and see if it's able to track it. So you can see it, well, unfortunately, was not able to track it. It's all empty. So we can track it manually for a few more. And let's see, maybe at this point it can track it by itself. There we go. So sometimes it will work really well, and the other time you just have to do it manually. Now let's say at this point right here we have a new cell that's being a new daughter, right? So we can click somewhere else so that we can see it, and then now we're able to select our new daughter. Right, let's see it grow. Let's see it grow. Right here. So you can see now this daughter is getting washed out, so we can say it's washed out. And let's say we made a mistake, right? Let's say now that it's budding, right, at this frame, let's say we are in this frame we say that we had a new daughter, which we didn't, but let's say for example we made a mistake. Well, if you want to remove a daughter from the list, all you have to do is just double click on the daughter and it will get removed from the list. And then if you want to save this information, meaning that this was all correct, then all you have to do is just retract the mother cell. So you just retract it. So you can see all of these are correct. We have the other daughters set up. And if you were to refresh the page, we still have both of our daughters, and we do not have the third daughter anymore. We were able to delete it. And if there are examples where you want to remove one instance instead of removing the entire daughter, maybe you want to remove, maybe at this point right here where it gets washed out, maybe you want to remove that and you want to say this was the frame that it was washed out. So let's say this was washed, right? It automatically removes any future daughter as well, right? So it has that capability. But if you want to personally remove it, all you have to do is just right click on the daughter itself, and now the daughter no longer exists for that frame. So there's multiple ways to approach it. You can either remove the entire daughter, you can remove one instance of the daughter in one frame, or you can change the status of the daughter for watched or died, and it will remove all the future instances with it. And this will wrap up all of the functionalities for tracking and prediction. Next, we will be looking at the visualization stage, which will start when you label a mother cell as dead. So like let's go down here. So once the mother cell dies, which is around this step right here, then we will receive an email letting us know that we have begun doing the morphology. And then we can see the morphology results in the final step. And now the last and final step before we move on to visualization is taking a look at the outputs for these movies. So here I have the list of our movies that we were just looking at. And you can see the last track which we analyzed is now in training, which means that it's been processed, it's been tracked, and now it's in the training phase, which means it can be used to train future machine learning models. We can also see for how many frames the mother cell was alive for, and we can see how many children it had. We can also take a look at this in the first trap for XY2, which we also tracked. You can see this one was alive for 258 frames and it had 3 children. So with that, we are now ready to look at the visualization stage. So once the mother cell is indicated that it has washed or died out, it means that the movie is finalized, its tracking is finalized, and you will receive this email saying that the morphology is now has been detected, and we're going to start processing it. Now, at this moment in time, you will receive this email, but we don't start processing until all the movies in the project their tracking has been finalized. But in the future, we're going to probably do it on a movie by movie basis, so that once you finish one movie, you can see the results immediately afterwards. But right now, you just have to do all the movies all at the same time for one project. Until then, then you can visualize all of the results at once. Once the movies have been analyzed and tracked, we get a CSV sheet, and now here we are mapping the CSV columns to the visualization data. So here we're setting up the position, the trap, the time, the directory, and the X and the Y. And now we see a blank page, so we need to go to the filters. And here we can see the different variables that we can manipulate. So we have X and Y and area and perimeter. As you'll see in a second, so when we click on that area, now we have our cells kind of appear here. And the X and the Y position are directly coming from the CSV. And the colors are coming from the variable we just chose, in this case, area. So now we're clicking next, which means we're moving through time. This is a temporal view. And as we move through time, we see the X and the Y are changing, but so are the colors. And you can see as the time passes, the cells start to go from one end of the color spectrum to the other, which indicates their area is growing in this case. And you can see some cells grow faster than others. And then if you want to see all cells at all times, of course, we can do that through the spatiotemporal visualizer. And here you can kind of track and see what is the X and the Y, what is the area for these cells, and the trajectory of growth until they die. And you can see that for those blue ones, those are the earlier time points. And then for the red ones, those are the later time points. And we can actually zoom in and track on one cell over all of its time points, which in this case, we call it the spatial view. It's only one cell, and we're looking at all of its viewpoint, all of its time points, all at the same time. And then in the temporal view, it's all cells at one time point. Additionally, we can change the colors as well. So here I am setting up a different color scheme to visualize. So now if we click refresh, now we can see the colors change. And now we're going from yellow to pink, pink indicating the largest area and yellow indicating the smallest area. And again, you can see the same thing. We're in the temporal view, and we're able to move across time points, and then we're in spatial temporal view, which we can see all cells, all time points. We can also limit the area as well. So in this case, we're going to limit it to from both ends, so we can look at cells above a certain area threshold and below a certain area threshold, and you can see those cells are now turned white, right? So all the other cells are the cells that that are within that threshold, and the white cells are the ones that are outside of that threshold. So now you can see in the temporal view, we can also do the same thing, right? So we've already gotten rid of a bunch of the cells, right? Because their area is below the threshold. And if we were to update this back to its original values, and now you can see all the cells are the right color. They're no longer white. Next up, we have the ability to select cells. So here we can draw over a specific cell cluster. As you'll see in a second, we can select them and view additional information or export their data. So here you can see we are selecting a second cluster. And now we're going to do a more complicated selection. So you can see now we're selecting a whole bunch of clusters all at the same time. Now this method is accurate for the most part, but there are some, as you can see, there are some other cells that are selected incorrectly. And so what we can do is we can remove them from our selection, these false positives, right? So once we remove them, now we only have the cells that we want. Now, sometimes you need to do it you need to be fairly accurate, and with a mouse, you might not get it right on the first try. So you can just keep drawing over it until you're able to get rid of all the cells that are false positive. And so now with our selection, you can actually see that we have 562 cells selected at various time points. And if we click it, you can actually see a list of all of them and be able to find the image for the cell as well as additional information such as extracting the CSV data for it. If you click on them, you can also see more information regarding the overall view from a graph standpoint of the perimeter over time, of the coordinates over time, as well as the area over time. And we can visualize that for this one particular cell or versus the average cell. And so you can kind of have that view as well, that zoomed out view. And this summarizes all of the main functionality of this product, going from all the way from uploading your data, segmenting the cells, tracking the cells and the mother cells and their daughters, and then visualizing their morphology and extracting that data out by selection of the particular cells you're interested in. Hopefully you found this helpful. And if you have any questions, let us know.